Howdy everyone, Mr. Jennings here for another Big Orange Biology FRQ review. This is FRQ practice number 2 that was sent out. This is a combination of different years' exams, as usual. Now this year, again, the 2020 FRQ AP exam is a unique situation for the COVID-19 situation, pandemic. But this video here, if you're watching this from after this point of recording, it's still just good FRQ review. This is designed for the FRQ Essential AP exam for 2020, the year of COVID. Okay, so let's talk about this questions. In the class, I assigned you 2 here. These are both publicly released exam questions, so there's nothing to share these. These are from a while back, but I'm just getting around to them now. Okay, so we have 2 questions here. They're both of the shorter exam variety. On the AP exam, there's usually 2 longs and several little short questions. On the reformat, there is typically 2 long and 4 shorts to make 6 FRQs. However, this year we're only having 1 long and 1 short. Both these questions are what we consider 4-point questions. So this here would mimic the similar style questioning that we would see from the AP exam for this year's little short question, but not 100% the same. So they're not 1-to-1. They changed with the format this year. They were already changing the exam questions to be a little bit more structured on the style questioning and the science practices they were putting in there. This year, these exam questions are 100% 1-to-1, but they get the same idea. So let's talk about the first one here. Now the first one in my class, I believe we assigned this as an AP test question. This was actually assigned to us in a regular classroom exam, I believe on Unit 4 or 5 exam, probably Unit 5. It's a good exam question. It's a great one for mutations and also ties in some Mendelian genetics. So this question here has 3 components. I'm going to kind of space it out here. Let's read through them. So in the prompt, the stem of the question, remember this here is the stimulus, so all the information you need really is in that prompt. It says gibberellin is the main primary plant hormone that produces stem elongation. GA3 beta hydroxylase, also known as GA3H, is the enzyme that catalyzes the reaction that converts a precursor of gibberellin to an active form of gibberellin. A mutation in the enzyme gene results in a short plant phenotype. When pure-breeding tall plants is crossed with a pure-breeding short plant, all offspring F1 generation are tall. When the F1 plants are crossed with each other, 75% of the F2 generation are tall and 25% of the plants are short. Okay, now the reason this part here is going to get to this part. Let's go ahead and dissect the stem just a little bit. So when it's saying this component right here, this little part here, when the pure-breeding tall plant is crossed with a pure-breeding short plant, all this is really telling you is that it's a simple, tall, short. And it's going a little bit further. If it's tall, it has gibberellin. If it's short, it has no or less gibberellin. Something's up there. Because if you read the previous part of the prompt, gibberellin is the primary plant hormone that promotes stem elongation. A mutation results in a short phenotype. So it says right here, the short phenotype is a mutation and it's recessive. It's a recessive mutation. Alright, so question A of this prompt. The wild-type genotype encodes for the GA3H enzyme with alanine, a non-polar amino acid at position 229. The mutant allele for the enzyme codes for the enzyme with threonine, which is THR, a polar amino acid at the same position of 229. So the question is asking you to describe, remember the bold is the action words. That's what you have to do. So first thing we have to do is be able to describe. So we're going to do that first. The effects of the mutation on the enzyme. Then we have to give reasoning. Sorry, I was about to put reasonings, but it's just reasoning. To support how this mutation results in a short plant phenotype in homozygous recessive plants. So the bold is always what you're actually doing. And these are the action words. And based on the number that comes after the bold text, it usually tells you how many points each action word's worth. In this case, there's no modifying number, so they're singular. So we're going to get +1 for doing the description and +1 for doing the reasoning. So to describe this mutation, we go from alanine to threonine. We go from a nonpolar to a polar. Now you can go and get really specific with this, because what we're doing is we're changing the amino acid structure. We're changing its tertiary alignment, because all of a sudden now we're changing its primary chain, which has an impact on its tertiary. If you don't remember your levels, let's kind of move over here to the margin. Let's go over here for a second. So just to kind of talk about the protein levels, remember you had your first level, which is your primary. When you talk about the primary level, that's just the amino acids linked. Amino acids to amino acids. That's your primary. Your secondary is basically the amino acid folding due to interactions between carboxyl and amine groups. Remember, the carboxyl and amine groups kind of are attracted to one another, so they're going to cause some folding. Into the tertiary, you're going to get more folding, and that's because of interactions with the R group. And I'm not going to go into the fourth level, but the fourth level is the interaction between R groups of different amino acid chains. So just keep that in mind. You could go ahead and be very specific here if you wished. I recommend, though, if you have the option to be kind of, not want to say vague, but if you can be broad and still get the full points, you may want to try to be broad unless you're very confident that you are 100% certain that you're being specific in the right way. You can remember the difference between secondary and tertiary. You may not want to say that. What you're better off saying is a change in amino acid structure results in a change of function. Alright, so let's go back to the question for a second. Just make sure we clear up some stuff. So the effect of the amino acid, the mutation on the enzyme. I need to put that there. So let's make sure I put that in there before I forget it. No, not substitution. So it's a change in amino acid structure due to alanine to threonine result in a change of function. Alright, I believe I ended up covering both in that statement. Alright, so the effect of mutation on the enzyme, it changes the structure. Provide reasoning how this mutation results in a short phenotype. Okay, so we talked about the change in structure results in a change of function. And we can go a little bit further to talk about how that affects the phenotype by saying this causes gibberellin. I'm going to abbreviate it for this purpose, but in the exam you would elongate it, right in the full form. This causes gibberellin to function less, thus producing less growth. Alright, so change in amino acid structure due to alanine to threonine results in a change in function. That's a description. Now the reason to support my description is this causes gibberellin to function less by producing less growth. And you can go a little bit further by saying as evident by the short phenotype in mutants. So you can talk about mutants as a short phenotype. Now this here is two parts, just to kind of clarify. We'll have, this here's I'm going to underline the box in the area with green. This here is really the description, and in red, this is the reasoning. So that's the description, that's the reasoning. Putting one without the other will only get you 1 point. Putting both will get you 2 points. Sometimes you can write one sentence and actually say both. I recommend making sure you are certain with that. Like, sometimes, some of y'all may feel like that first statement does both, but we need to be more accurate by saying the exact situation. We're talking about gibberellin, so you want to make sure you are more specific to that. Alright, question prompt B asks you, predict the change in the codon sequence that resulted in the substitution of alanine for threonine at amino acid for 229. Okay, for this one, you just had to look at what the amino acid codon for alanine is. So alanine's amino acid codon is right here, and it seems to be G, C, and it doesn't matter what the third letter is, so we usually represent that by N. N is, you just put N on your thing, you don't have to explain what it is, but if you feel comfortable doing that, you can. All N is the amino acid. This is just a general and nitrogenase works fine. But we're going to say general nucleotide base. This is generic nucleotide. So if I use this the end situation here, it's just generic nucleotide. So we go from alanine, which is GCN, it could be UC or A or G, to threonine. And threonine is above it just a little bit. And we can see here, threonine is ACN. So what we can do for our answer, you put this information, then you would put this for your answer. Because you have to predict. You would say the change of guanine, or just G, in the first base of the codon to A, also known as this change, you could be phrase it as the substitution, if you want to get a little bit more accurate. So the substitution of guanine in the first base of the codon to A, or adenosine results in a change of amino acid. Pretty straightforward. That one, by the way, was also 1 point, because all it was asking you to do was make one prediction. This here is merely an evidence piece. You do not necessarily have to write it, because you can just write what I wrote here and it would be fine. But having this here kind of explains to the reader that you understand what you're saying, because you're showing the similarities and the differences at the exact same time. Alright, C. You're to describe how individuals with one heterozygous or two homozygous copies of the wild type GA3H allele can have the same phenotype. Basically, it comes down to like this. Phenotypes, the alleles are all about basically a chromosome. When you have two chromosomes, I'm going to do it over here on the sides, just so I can show you. Sorry, downloaded, finished. So let's say we have two chromosomes. Two homologous pair of chromosomes. And this one here, this is the locus for the GA3H gene. In a dominant situation, which is going to be just a normal situation, it'd be like this. But in a mutant, I'm going to draw a little bit shorter for space, it may look like that. And this here again being the mutant. What happens is, and this is really going to be the answer, there is enough gibberellin, or GAH3 enzyme, produced by the one normal allele, or we can call it a wild type. Under normal or wild type situations, it doesn't matter that this one's defective, this gene here still produces enough GH3 enzyme to go make gibberellin. Okay, enough of that enzyme is being produced regardless of the fact that there's only one copy of the allele, but one copy of the allele is still doing enough work to make this functional. That's not always the case sometimes. In some really weird situations when you have co-dominance and incomplete dominance, but that's how heterozygous individuals are still dominant. That's what I mean by dominant. The dominant one still produces enough of that enzyme, it functions no matter what, essentially. The recessive individual, like the homozygous recessive, just doesn't have any of that enzyme's coding gene to actually do the job. Now, that's under simplistic Mendelian term, but that still works just fine. Alright, that one's also 1 point, by the way. That was, again, another +1 question. I meant to say that, because the only thing you were doing was providing an explanation. Alright. Okay. So, next prompt. Next in here, genetically modified crops have been developed to produce a protein that makes the plant resistant to insect pests. Other GMOs, genetic modifications, make the crops more resistant to chemicals that kill plants known as herbicides. A. Predict two potential risks of large-scale cultivation and use of genetically modified plants. Okay, so this is an example here where you have one action word but a modifier. That means this question here is 2 points. Alright, so you got to predict two potential risks. So there's a whole laundry list the reader's guide gives you. I'm just going to go with a couple. First off, the biggest risk is you're going to have a lack of genetic diversity is a risk with a monoculture of one crop. So that's a big risk. And another risk could be, by the way, don't do bullet to list. So I probably should not do that. So I want to bullet it. So lack of genetic diversity is a risk with monoculture of one crop. Additionally, with the little transitional phrase there. Additionally, if there is a disruption to the insects, this could destabilize the food chain, because the insects that eat those crops are now all of a sudden not getting the food they need. That can be disruptive. You can also create a selective pressure where you promote resistance within the crops. You can also create a situation where you inadvertently kill plants that you weren't trying to kill with herbicides. There's a long list, but these are two good ones. So, one, you have a lack of genetic diversity. Two, there is a disruption within the food chain. Now, the solutions. And now, this is where this one doesn't have a modifier, but it's assuming you can read the lines here. So for part A, you have to propose a practical approach for reducing the risk for each. That means you need to have a risk-reducing factor. So B, genetic diversity could be improved by interspersing, fancy word here, I misspelled interspersing, but whatever, interspersing wild types with the GMO. So when you have a GMO plant, if you alternate with a wild type in there, that wild type will provide genetic diversity to the ecosystem. Now this causes another issue, possibly with the spillover of the genes. But what that means also, though, is it makes it less likely for disease to spread. Disease in plants spread by local transmission, either through soil or through airborne pheromones and other airborne chemical spread, or spores or whatever, depends on the disease. That's really the main risk of being a monoculture. So, if you were to reduce that by reducing the amount of individuals that can transmit the disease with a wild type, that's how you can do that. Genetic diversity can be improved by interspersing. As for the food chain can be stabilized by providing alternate food sources. So if you're worried about your food chain being destabilized because of you just introduced a pest-resistant plant, providing alternate food sources for those pests to feed off of will keep the food chain stable. That goes back into the interspersing with wild types. You're not going to harvest the wild types more likely. The wild types are just going to basically be a buffer, and they could also be keep the food source alive. The problem with that is still, though, there's a spillover issue that could happen. So that's something to keep in mind. Alright, but this here was, first part here was 1 point each, because you actually do this separately. So if you can only come up with one risk, that's still a point. And if you can only come up with one mitigation for that risk, that's only a point. That's a point as well. So you can still get half credit by just coming up with one risk and one mitigation strategy, or 2 points for just coming up with risks. Now, obviously, you can't really answer B without answering A, so you can't skip A and just give some B answers, because obviously if you're answering to B, it means you have answers for A. But you can get some partial credit in all these. It's always beneficial if you are taking the AP exam and you're doing the FRQs to put something down better than nothing, and also, you'd be surprised how often certain things tend to show up. Structure and function of the protein, like we saw in the first question today, is a common theme that you may get lucky by putting that. Being able to identify mutations or just coming up with predicting of changes and disruptions is a very valuable skill, especially on this year's AP exam. Alright, but that's it for today. I'm Mr. Jennings. I hope you have a great one. See ya.